Year 11, this tutorial will focus on sections 2.3 and 2.4 of the AQA Biology specification. So we'll focus on photosynthesis and biology and organisms and the environment. So if we start off with what photosynthesis is, well, photosynthesis is simply a process that is carried out by plants that produces food for the plant. Now in this case, the food is glucose. Now essentially what happens is photosynthesis takes place in the leaves in an organelle called the chloroplast. Now the chloroplast contains chlorophyll, which is a green pigment which absorbs the sunlight. Okay, so it takes in this sunlight. Okay, now that sunlight is used to combine water and carbon dioxide. The atoms are rearranged and we get glucose, which is the food for the plant, created, and we also get this byproduct, oxygen, which, as you know, we use for respiration. Now, leaves are adapted to absorb sunlight for photosynthesis. So to start with, they have an incredibly large surface area. Okay, so that maximises the sunlight that they can absorb. But if we have a look a little bit deeper at the cellular structure of the tissue, we can see a number of different layers, and I'll go through a couple of the layers and what they do. So we've got the waxy cuticle, which is basically a protective layer, and it's waterproof. Okay. We then have the upper epidermis. Now the upper epidermis is transparent, and what that does is it basically allows light to get through to this layer here. Now this layer is called the palisade layer. Now if you have a look at this layer, you can see that the cells are really long. Okay? Look again. What else can you see? We should be able to see that they are packed full of chloroplasts. Okay? And remember, those chloroplasts absorb sunlight for photosynthesis. So this layer is for photosynthesis. Okay, then if we look at the next layer, again, it's all adapted for photosynthesis. We've got the spongy mesophyll layer. Now, the spongy mesophyll layer, you notice we've got lots and lots of gaps, and this layer is all to do with gas exchange, so swapping carbon dioxide for water. We then got the lower epidermis, which is also a transparent layer. We have two, in this diagram at least, we've got two guard cells which basically control the stomata, and the stomata is just simply a pore that allows gases to go in and out. So for example, in the case of photosynthesis, CO2 would be going in and oxygen would be coming out. A typical question that you might get in an exam could be something like, describe and explain how the leaves are adapted for the leaves. For photosynthesis. And you could look at each layer in turn and describe how it's adapted and how that helps with photosynthesis. Okay, so just to remind you about photosynthesis, so the reactants in photosynthesis are carbon dioxide and water. In the presence of sunlight, which is trapped by chlorophyll, we get the products, which are oxygen and glucose. Now, some of this glucose is used by the plant, and we'll come into where it might be used in a second. and some of it is converted into starch for storage. Okay, because starch is insoluble. So we know that glucose is the food that's made in photosynthesis. Now glucose can be used by the plant to create a number of things. Well, to start with, it can be used to create something called cellulose, which strengthens cell walls. It can be combined with nitrates to make proteins, and proteins might include enzymes and chlorophyll. Okay, so glucose plus nitrates equal proteins, and it can be stored as insoluble products. So this might include starch, which can be used for energy, fats and oils, and of course glucose can also react with oxygen to generate energy in the process of respiration. Now there's an experiment that you might get questions on in the exam, and this experiment is basically an experiment that we can use to prove that sunlight is needed for photosynthesis. So what we do is we take a leaf and we would de-starch it, so make sure all the starch had gone out of the leaf. Now remember, glucose is stored as starch. And then we would take a piece of paper that basically is opaque, so no light can get through it, and we clip that onto the plant. We'd then leave the plant in the sunlight, and then 3 days later we would decolorise and treat the leaf with iodine. Now iodine detects any starch, so if the plant has been photosynthesising, it should have produced glucose, and that glucose should have produced starch, should have been converted into starch. Okay, so what we can see here is only the parts that were exposed to the light have photosynthesised and produced glucose, which was converted into starch. And we can tell that starch is there because when we tested it with iodine solution, it turned black. Now, there are 3 factors that can limit or slow down the rate of photosynthesis: light intensity, carbon dioxide concentration and temperature. And what you might have to be able to do in the exam is you might have to describe how each of these factors affect the rate of photosynthesis. So if we look at this first one, we can see that as you increase the light intensity, the rate of photosynthesis increases until it gets to a point. Okay, and then we've got other factors that are limiting it. Again, so if we look at the graphs in turn, we've got this one, which is to do with temperature. As you increase temperature, the rate of photosynthesis increases until it gets to a point where the enzymes that catalyse the reaction are destroyed. Therefore, the rate of reaction falls. Remember, we've also got carbon dioxide that's a limiting factor in photosynthesis. You can see if we increase the level of carbon dioxide, the rate of photosynthesis increases until it gets to a certain point where we need to increase one of the other factors. So in this case, we need to either increase the light or the temperature. Now, if we were to do that, what we would see happen is that we'd get a line that looks something like this until another factor was limiting it. Okay? The final one is light. So as we increase the light intensity, so I'm reading this axis, the rate of photosynthesis increases until it gets to a point whereas it doesn't matter if we increase the light intensity, the rate of photosynthesis isn't going to increase any further. So what we need to do is either increase the carbon dioxide or the temperature because they are the limiting factor. Now, farmers can use their knowledge of limiting factors to increase their crop yields. This is because bigger crop yields equals more money. Okay? So we might put artificial light, okay, because remember there's only certain parts of the day where it's sunny, so this extends the time for photosynthesis. We might use artificial light in the winter when the days are shorter. Heating, okay, will increase the rate of photosynthesis, remember up to a point. If it's too hot, remember it will denature the enzymes and therefore the rate will slow down. So it's a very, very careful balance. Now, obviously this is really useful for a farmer when it's really cold, so he can grow his crops in the winter months. And adding carbon dioxide will also increase the rate. Now, what the farmer has to think about though is economics. So he needs to balance the cost of providing extra light, heating or carbon dioxide against the crop yield. So you might be asked to evaluate whether it is worth doing these things. So what's the benefit of it, but what might be the disadvantage? Okay. Now, one thing to note is a paraffin lamp will provide light, heat and CO2 all at once. So if we then look at the second part of this, so this is 2.4, okay, where we're looking at organisms in the environment. Now, the distribution of organisms, so if you imagine plants on a field, on the football field, or within a habitat, might be affected by a number of physical factors. So it might be affected by temperature, light, water, nutrients, the amount of oxygen available for respiration, or the amount of carbon dioxide. Now we might want to have a look at the distribution, so maybe how many daisies there were growing on a particular field, and we might compare that with how many daisies there were growing on another field and then try and work out why that might be. So maybe one field has got more light, or maybe one field isn't trampled on as much as another field. So one way that we can estimate a population size is we can use a sampling square called a quadrat. So this is a square frame that can be divided up into a grid. And then what we can do is we can count the percentage. Well, we can do two things. We can either work out how many of these plants are in this square. So we can say there are 1, 2, 3, 4, 5, 6. We're not going to count this one because it's halfway out, or you could say only count the ones that are more than halfway into the square. So in this one we've got 1, 2, 3, 4, 5, 6. We can then randomly distribute the quadrat in different areas, and we can do this using coordinates. Okay, so for example, if this was our field, we could have a tape here, so 4, 5, 6, and we could have a tape running down here. Okay. Use a random number generator to generate our coordinates, and then we could say, okay, we'll go 6 and 2, and that is where we would place our quadrat. We'd count the number of daisies within that quadrat. We'd do it maybe 10 times, so it's more reliable, and then we'd work out the mean number of daisies. Always use it to estimate the population size if we knew the size of the field. Another way we could do this is we could estimate the percentage cover. Okay, so we've got 1, 2, 3, 4, 5 squares by 1, 2, 3, 4, 5, so that's 25 squares, and we think we have daisies in 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 14 squares, so we'd say 14 out of 25 as a percentage. Again, we would repeat this. Okay, we don't just have to use random sampling, we can also use a type of sampling called systematic. And this is where you might be looking at changes between 2 points. So we take a tape, which is called a transect, and we tape it along between 2 points, and then at intervals along that point we would place our quadrat and we would count the number of organisms in the quadrat, and we might be looking at the different types of organisms that are present at each stage along the quadrat to see if, along the transect to see if it changes. Okay, so we take our tape measure, we lay it out, we place quadrats at intervals, so say maybe every metre, and then we might count the number of organisms at each station. We might use a key to work out what species of organisms we have and how many we have at each station. We might even take the soil pH at each station. We might take a reading of light intensity to see if that is the thing that's affecting the distribution of the organisms.